It's one of those days.
These are not the only person in the class this week.
What is it?
Not last Friday, there was one other person, but the Friday before I was the only person. Friday before that, I was the only person.
But there's a lot of people online on Friday this morning, so hey, that's awesome. Thank you for being awake, everybody. And if you're watching this video, no offense, I'm not talking bad about you, but thank you for everybody who's awake this Friday.
Hold that there.
Well, I guess I'll do announcements, reminders. The first thing to remind you is independent work. You know, when it started, that was a really easy assignment when you think about it, because you needed about 7 points a week. That's easy to do. But now, at this point in the semester, if you haven't done much, I'll admit it's hard to get that many points, because now you're way behind. But, you know, the sooner you start, the better it'll be. The later you start, it's just going to get so much harder and harder and harder. Every day that you wait to start, it's going to be harder and harder. Also, I want to address, we've got one student, I love this. I hope he doesn't feel bad, but he sends a lot of stuff and there's a lot of mistakes on it. But this is why I love independent work, is because, you know, it doesn't matter how many mistakes you make, because you can keep turning stuff in until you hit 100. But anyway, one of the reasons I love independent work, like I was getting at, is it's a great conversation for us to have, even though we're not speaking. It's like you can turn something in and then I can write back like, yeah, that's great, but did you consider that your source is crap? Or, you know, did you really read this? And, you know, it may come off as harsh because we're not speaking, it's all in writing, but I just want you to know, when you get those kind of things back from me, I'm not being harsh. It's just a great learning moment. It's a great way for me to say, hey, you know, you're not really comprehending what you're reading, or, you know, you're not really paying attention to your source. Things like that. So again, independent work on my end, that's why I love it, because it's there for me to help you learn, and not just learn biology, but more importantly, like learn how to come up with a question and find the answer online and be able to weed out all the bad answers that you find. So, you know, you can Google anything and find the answer to anything. Any answer you want, you can probably find it online. So it's important to be able to look up and get the truth. And I hope that I can help you do that. On my end, that's why I love it. On your end, remember, that's a free 100 points. This is a 1,000 point course. So 100 points is a great letter grade. So if you say you're not bad at math, or you're not good at testing, or you're not good at science, or whatever it is, keep that in mind. You have that in your control. Doesn't matter how bad you are at testing, or how bad you are at biology or science. That's 100 points. It's 100% in your control. So, you know, get to it. Let me help you if you need to. Also, sorry, I'm going off on a tangent. Speaking of which, if you say, oh, I'm bad at testing, and like I just said, you should do independent work. But on the same note, if you say you're bad at testing, then do the study guides I provided you. Because that's how I write the exams. I take those questions, I reword them, and then I give them to you. So not only will you be getting independent work points, but you'll be studying the questions that you will see for the exam. So any questions about independent work? All right. Just trying to help you. And I know for a lot of you, especially those of you who are doing good, you're probably just like, just shut up about it already. Did you get tired of me saying this? I've just been doing this for a while, and I know that a large majority of the students who just don't seem to care right now are all of a sudden going to care a lot in the last week or two of class, and then they're going to get mad at me when I say, there's nothing we can do, because I've been asking you all semester. Anywho, if there's no questions about that, oh yeah, the other last announcement is I have a department meeting today at 10. So from 9 to 10, my office hours will be physically in my office, but then after that, I'll be in the meeting, but you can still meet me online. So if you need me, I'll still be online. I'll just step out from my department meeting if you show up online. So any questions about that? All right, let's talk about mutations. So this is really important stuff. And we're only going to scratch the surface again, because it's a 100 level course for non-biology majors, but this is really important. So for example, when people say, you know, we have all these new variants of COVID, right? And you're going to learn how that happens.
I don't mean to interrupt you, but is there any way you can put the presentation up? I can't see the slides.
Yes, that's the second time I've made that mistake. And thank you.
Sir.
This time you told me right away. I'm going to give you a little extra credit on your attendance for that, Matthew. Thank you. Come on, where you at? Bear with me, sorry. Better to go through this now, though, than later. Slideshow. Slide. There's that. That's still up there. And there we go. Good? Hope so. Anyway, sorry about that, everybody. And I'm glad Matt told us early rather than later. So anyway, when we're talking about COVID, we have all these new variants of COVID, and the way that happens is from mutations. So to understand how that works, first thing you need to understand is how mutations work. Then, in the next chapter, as you already know from the lab, we are going to learn about evolution and also natural selection. And you're going to learn that without mutations, there would be no evolution. Without mutations, there would not be that one first black moth, right? So there was just a bunch of light-colored moths, and then randomly, and it maybe had something to do with smoke, maybe not, we don't know that. But randomly, there just happened to be this light moth that had a mutation where it makes eggs, and it laid eggs, and then we had a few black moths that were born, right? It was completely random. It was due to a mutation. It just so happens that that mutation gave them an advantage because everything was dark because of pollution, but that's a story we'll get into chapter. But yes, all this stuff is very important. What is a mutation? I'm not going to ask you the definition of it, but the definition from your book is a mutation is any change in the nucleotide sequence of DNA. So that's what a mutation is. So prior to taking this class, you might think of a mutation like from The Simpsons, where they have that three-eyed fish. You know, a lot of times when people think of mutation, they think of like what it looks like, the phenotype, if you will. And yes, sometimes a mutation will lead to something that looks different. You can look that up too. A lot of times there's things that are huge changes, right? So there might be one small mutation which leads to completely not having limbs or having completely extra limbs. And in the story of natural selection and evolution, that's actually a really big thing. Because sometimes change isn't small and sequential. Sometimes it's just one big step. But again, that's a story for next chapter. Mutations can involve large regions of chromosomes. So remember when we're talking about crossing over, what I didn't say back then was sometimes things go wrong. Sometimes you could be missing a complete huge chunk. Sometimes you get a bigger chunk. Sometimes things get copied, and we don't even get into that. Sometimes like you get a chunk that's supposed to be like that big, and then there's just like multiple exact copies of that one chunk of chromosome. We don't even talk about it. It's so fascinating, but we don't have time to talk about it. The mutation can also involve a single nucleotide. So you might swap out the letters. Remember the letters are A, T, C, and G if we're talking about DNA. So it might be instead of having an A, you have a C, for example, right? You might swap them out. So again, we're saying it can be very huge, like huge chunks of chromosomes, or just a single nucleotide pair. And as that third one says, anywhere in between. And between will be the first attendance word. Between. The next slide I'm going to show you is this is kind of a decent picture. This picture here is not from your textbook, but it is a picture of a single nucleotide pair, right? So here, the original letter is a T, but then by some mistake, probably by DNA polymerase, like when it was supposed to make the perfect copy of the DNA, and maybe brought in the wrong nucleotides, and now we have a C instead of a T. And then let me show you this picture before I ask if there's any questions. But you can see here, and this isn't just some random example. This is a real life example. This is what sickle cell is. So we zoomed into this gene. The gene's really long, but we're just looking at this one codon. Remember, a codon's a word, right? So this one codon, this one word is supposed to say CTT, right? Which leads to the mRNA having the code GAA, which tells the ribosomes and the tRNAs, hey, bring me the GLU amino acid. But instead, for some reason, this one T has been substituted for that A, right? Just this one letter. And then because of that, on the RNA, we have a U instead of an A. And then because of that, instead of having a GLU amino acid, we have a VAL amino acid. And that might not seem much, but as you already know, because we've hinted at this earlier, sickle cell is a pretty big disease. And just having that one letter and then that one amino acid off means the whole protein is now misshaped. And now, because the whole protein is misshaped, the whole red blood cell is misshaped. And Now, because of that, you have all this slew of problems that we talked about in the previous semester, or previous chapters. So this whole disease is caused by that one switching of the letter, right? So it can be a big deal, even when it's just one letter. Anyway, that's just an example. I'm not going to ask you any questions about sickle cell. Here's what you do need to know for the exam. There are two categories of mutations, and you need to know the differences between them. There are substitutions, and then there are deletions/insertions. So the one I just showed you was a substitution, right? Instead of a T, it was a C, or whatever it was. Again, it's just an example. You don't need to memorize it. But there are substitutions, and there are deletions/insertions. There's also other ways of categorizing it, and we'll talk about that later. So if you get a question on this, it's not like I'm going to say, what are the two categories of mutations? It'll be more like, you'll need to understand the difference between substitutions versus deletions/insertions. Yes?
So when we use mutations, do you have to be like born with it, or can it like?
That's a great question.
Be like grown?
So she's asking basically like to have a mutation, are you born with it or other? And I think we may talk about this later in more detail if I remember the PowerPoint correctly, but it's kind of both. For example, if you were born without any mutations, then you're just, you're normal. Well, all your cells are normal from that point of view. But if you get, exposed to too much X-ray, too many UV lights, or any other carcinogen that you may eat or drink or smoke, then you might have a mutation. And if it's a mutation in your arm, maybe it'll be cancer, maybe it'll kill you, maybe it won't, whatever. But if it's a mutation in your ovaries, where you're making eggs, then that mutation may be something that's permanent if it gets passed along to your kids, because then your kids will have that mutation. Does that make sense? So if it's a mutation anywhere on your body, in any of your somatic cells, it's just your mutation. But if it's a mutation in your reproductive cells, and then those cells are used to produce a baby, then that mutation goes on and on and on.
Okay, so that's why they ask you heart disease or anybody in your family had breast cancer before.
Oh yeah, definitely the breast cancer thing.
That's why you might be at a higher risk because of that mutation.
Yeah.
I don't know why.
Yeah. The breast cancer is a great example of that. Because we've talked about what cancer is, and it's basically a disease of the cell cycle. So the on-off switches aren't working. So there's something wrong, and there's a particular gene. You should know this. I can't think of it right now. I don't want to waste your time thinking of it. But there's a certain gene that if you have a mutation, you're at a higher risk of having that breast cancer. And that mutation probably came from your family. Of course, you could have been the first to get it, but we don't know. Yes.
Can it skip generations? So for example, my grandma had breast cancer, my aunt had breast cancer, but my dad doesn't have the gene for any type of cancer. And then my other aunt doesn't have the gene for the cancer.
Yes and no. So for those of you online, if you can't hear the question, can it skip generations? It all depends, and this is a great question because it goes back to the stuff we talked about when we talked about crossing over, independent assortment, segregation, right? So if your grandmother has a mutation on one of her chromosomes, and then she produces eggs, or produced, right? She's grandma, so she might not be producing eggs anymore. But when she produced her eggs, and if she only had one mutation, one chromosome was mutated and the other one wasn't, then half of her eggs would be completely normal, and the other half wouldn't, right? So then half of her kids, statistically would have the mutation, and half of them wouldn't. If that makes sense. It all depends on which egg you get, if you get the mutated one or not. And then, even on that note, let's say your mom, we're talking about the right grandmother, whatever. If your mom was the one that got the mutated chromosome from your grandmother, but she also got a normal version from her grandfather, she too is, again, heterozygous. So when she makes eggs, she too could pass on a normal one or a good one. So yes, a long story short, yes, it could skip generations that way. The same way any phenotype we've talked about, or genotype, can skip generations. Because like we talked about, the purple allele, the first thing I told you when we learned about this was, a purebred white flower and a purebred purple flower, they mate, and all the flowers are purple, but then you mate those, and then some of them are white again. And yeah, because the white trait skipped a generation, and the mutation is essentially just a trait. So everything you learned about that, everything you learned about genetics is the same here. Because technically, that is a mutation. Everything that's different, all these different alleles, that's how they came to be anyway, it was a mutation. They used to be all nothing but purple plants, and then at one point, sometime in the history of those plants, there was a mutation, and then all of a sudden, boom, you have this new phenotype. It's a really amazing book. I'm going to tell you about it in the next chapter. It's more about natural selection, but again, without this, natural selection wouldn't be a thing. But great questions. Anything else? All right. So again, you need to know the difference between substitutions and insertions/deletions. So sickle cell was a great example of a substitution. Here's another one. So instead of a G in this gene, right, we've substituted with an A. And because of that, we have a completely different amino acid. And again, like we talked about from the sickle cell, that could be a bad thing, right? If you have one amino acid that's different, then the protein might have a different shape, and that might be completely disastrous. Of course, it might not be a big deal. Maybe it helps you. Maybe something like that makes you go from a purple flower to a white flower. I don't know. But the point is, that's a substitution, right? You can see by the substitution, we have only changed one amino acid. I would say, as far as the exam is concerned, that's the most important thing to focus on. When we substitute this one base, right? That's the only thing we did. We just changed something. We just changed one base. We substituted it. Notice the result is a change in one amino acid. Excuse me. Yes, one amino acid. And relatively speaking, as you're about to see, that's not that big of a deal. I'm about to show you a deletion. But are there any questions about substitutions before we talk about deletions? All right. Picture, if you will, what we're actually looking at is a gene, right? So this would actually be really, 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 really long. Like if we were to somehow write this all out, I'm sure it would go all around this room a few times, right? Because genes are long, they have a lot of codes. But we're just zoomed in, right? So we've zoomed into this gene. We're looking right here. And where it's supposed to be a U, this is a bigger mistake. Now, instead of swapping the U for something else, the RNA polymerase just completely messed it up, and the U is just gone now. So it was deleted. So now, as far as the exam is concerned, this is what's important. Now we don't just have one new amino acid. Now everything down here, and right now we can only see a little bit of it, but imagine it keeps going. Now everything from that point down is now mutated, right? We haven't just changed one amino acid. We've changed, depending on where it is, maybe thousands of amino acids. Everything from this point down is now a new amino acid. And hopefully you can see how that happens, right? Imagine, before I ask you or take your question, imagine like physically, imagine what would happen if I were to like just pull this out, right? And everything just went and shifted to the left. Can you picture that? Like if you could physically grab it and pop it out. Maybe if it was at an angle like this, so gravity's making all these things like stack down here, and I pop it out and everything will just go and shift to the left. Can you picture that in your mind? Because that's what's happened. We pulled out this U and everything shifted left.
How was the U deleted? It just.
There was a mistake. So, so RNA polymerase is reading the code and, it reads it, knows it needs to bring in a U, and for whatever reason it didn't. Maybe there just wasn't a U available, so we just kept going. And as we learned when we talked about RNA polymerase and transcription, we learned it's very accurate and very fast. But when I said very accurate, I also gave you some numbers that you didn't need to know. The point is, despite the fact that it's very accurate, it is not foolproof.
So it can function without missing one. It doesn't need to be full.
It could, yeah. And that's where mutations come from. In theory, the way it's supposed to work is it recognizes when it makes a mistake. And we didn't really get into this much, but the polymerase recognizes when it makes a mistake and backs up. It'll literally back up and fix it. Also, it's worth noting that's not always the way these mutations happen. It doesn't have to be in the copying process. This mutation could have happened because you're smoking too many cigarettes, right? There's carcinogens in cigarette smoke. So that might be, it hits your cells, and that might be that was knocked out. Or UV light, right? Maybe you're getting too much sunlight, and all that UV energy hit your DNA right there at that spot and knocked out that U, or your RNA in that point.
Can it delete more than one, or is it just one?
Great question. Yes, it could delete more than one. And we didn't even talk about that. But yeah, it could delete more than one. And here's the thing too, even just one, That's the point, one of the points here. Even if you just move the one, it's so disastrous because again, it's not like we just changed this amino acid for this amino acid. Now everything downstream is now messed up.
So if you move one from the very front, it messes up the whole chain.
Yes. Yeah. Of course, then again, if you did it in the very back, yeah, it would essentially be almost like a substitution, almost. But again, 100 level class, we don't even get into that as far as exams are concerned. But again, speaking of exams, the big difference here between a substitution and an insertion, because obviously that in itself is easy. A fifth grader can get that. So are you substituting it or are you deleting it? That's easy. But the thing you need to keep in mind is if it's a substitution, we change one phenotype. One amino acid. If it's a deletion, we change a huge chunk. Because, and I'm going to show you this one more time and then we'll move on. And you can see this is what we're reading, right? It's the triplets, right? Those are the codons, those are the words. So that's a word, that's a word, that's a word, that's a word, so on and so forth. And it still happens that way. Those are still the words. We're still doing it by 3. But now, because we lost this U, what used to be a UUU was now a UUG, right? Because this thing switched over. And now what used to be a GGC is now a GCG because everything switched over. Anyway, that's a deletion. Are there any questions about deletions? Insertions are pretty much the same situation, same problem. As far as the exam is concerned, in a way, you can almost consider insertions the same as deletions in that they're disastrous because when you put the one extra in, then everything downstream is now messed up. So we're not just substituting one amino acid for another. Now everything down this way is messed up. And again, this is only 1, 2, 3, 4, 5 codons. And remember, there's thousands of codons in a gene. So we're just zoomed in on this one part. So this mistake could be huge. So are there any questions about insertions? All right. There's yet another way that we can categorize and differentiate mutations. And one of these is going to be the next attendance word. That will be nonsense, will be the next attendance word. Anyway, so not only are you dealing with, is it a substitution or is it insertion/deletion, right? Those are 2 categories. Another completely different way of looking at it is it a missense or is it a nonsense mutation? So let's talk about the difference. And that is a little bit tricky, right? Missense, nonsense. What is this nonsense? A missense mutation is a mutation that changes the amino acid coding. So far, every example I've given you has been a missense mutation. Because now we have a different amino acid than we were supposed to. At least one. Right? And in the case of insertions and deletions, many amino acids. But that's missense, right? That's everything I've just shown you is a missense mutation. The other thing would be a nonsense mutation. That one's a lot easier. Nonsense just changes whatever code it was to a stop codon. Remember we told you that there were 64 triplets and only 61 of those say, hey, bring me this amino acid, bring me that amino acid. Because some of those just say, stop, the process is done. So if there's a mutation that said, bring me this amino acid, and instead you change it to say, stop the process, that's what a nonsense mutation is. And think about the ramifications of that. In a sense, the nonsense mutation is bad, if you want to call it that, as an insertion deletion. Because now you're not just changing one amino acid for another, you've messed everything up down the line. Of course, if that happened at the very end, no big deal. Or like she asked earlier, if it happened at the very beginning, then you've just essentially deleted the whole gene, right? So yeah, missense versus nonsense. Make sure you know that. There's also a third. I think I have it on here, so I'm just going to bring it up just in case I don't, because I think I have it in writing. It should be here. But then there's silent mutations. So a silent mutation would be you change one of the letters, like I showed you earlier, a substitution. But even though you changed one of the letters, which changes the word, remember some of those words code for the same amino acid. So you could change a codon, but still have the same amino acid. And I wish I had my little wheel up. Maybe I'll have it up later, where I can show you like this equals that amino acid, but so does this. But we'll come back to that. For now, just focus on missense versus nonsense. Also, another vocabulary word for you, if you will. We talked about insertions/deletions. Those are called frame shift mutations. So now we have missense versus nonsense versus frame shift mutations. And like I said, they often have disastrous effects, because we're no longer just changing one amino acid, we're changing everything down the line. So any questions about this slide? The next slide I'm going to show you is another picture of an insertion or a deletion, I'm not sure which. Either way, it's a frame shift mutation. And this picture is not from your book, so I just wanted to give you a different visualization. It shows the same thing, just shows it slightly different. So which one did we do? Well, yeah, so, because something was inserted or deleted, we're not even focused on that. We're just saying because of that, something being just inserted or deleted, what we have is a frame shift. So instead of CAT, TCA, CAG, right, those letters are still there, something happened down the line and now we've just shifted. So what used to be a CAT is now ATT, right? And what used to be TCA is now CAG, right? So you just completely messed everything up by inserting or deleting that one thing. So again, I've already shown you this. This is just a different way of looking at it. That picture's not from your textbook. And here's a handy little chart that you should study, because it's everything we talked about, but in one chart. The type of mutation. There's substitutions versus insertions or deletions. Right? And under substitutions, we have, there it is. That's the one I was telling you about. For the substitution, you could have a silent mutation. Right? So yeah, you change the letter, which means you change the word. Sometimes the word codes for the same amino acid, in which case it doesn't even make a difference. And then of course there's the missense, which we've already talked about, and then the nonsense, which we already talked about, to stop the whole process. And there's a question on the study guide that'll probably be on the exam. It's basically like, what would happen if there was a nonsense mutation? And one of the choices is there would be a shorter polypeptide, and that is the choice, right? Because what we're saying here is you've changed one amino acid, or one code that used to be for amino acid, it now says stop. So obviously the answer to that would be, there would be a shorter polypeptide. And then of course, like we said, for those insertions or deletions, those are called frame shift mutations, like we just talked about. Those are very bad, generally speaking, because it's like essentially you have a whole new gene, a whole new protein, which sometimes can be good, as we'll learn when we talk about evolution and natural selection. All right. So now that you know the different types of mutations, let's go back and talk about what we've already talked about before. How do these mutations how do they happen? So let's officially talk about it. Mutations occur in a number of ways. The first one that I've been talking about a lot is error in DNA replication or recombination. In other words, again, when that DNA polymerase is reading the code and it's trying to make a new strand of the DNA and it messes it up. So it doesn't make a perfect copy anymore. It's trying to read that original strand and messes it up. So now the new strand has a mutation. That's one way. And when we say recombination, we're talking about crossing over, right? So sometimes when you cross over, things don't happen exactly where you need them to happen, which makes sense, right? That's just a chunk of chromosomes being swapped. So you can see how mistakes might be made. And then there's this other stuff, and this is what I was getting at earlier. There's physical and chemical agents called mutagens. You should know what a mutagen is, what it is. We've been talking about it, and now I'm giving you a name for it. The mutagen, those are the things that cause mutations. The most common physical mutagen is high energy radiation, X-rays, and ultraviolet light. I'm not going to test you on that, so I'm going to put it next to it, but obviously that's good information for you to know. Then there's also chemical mutagens. I'm not going to ask you about any of this, but it's definitely worth knowing, right? Because this could be life or death for you. So this is also really interesting independent work stuff, in my opinion, if you want to look into it. Think about Chernobyl, right? Huge nuclear disaster, a lot of radiation. Things are still alive there, and humans are now allowed to go back in and do some studies. So I'd be interested to see, I'm sure people have done studies, what are some of the mutations that have happened. But anyway, yeah. any book says, because of these, some of these things that cause cancer, you should avoid them. But yeah, there's a lot, and also chemical mutagens. That I would really look into that one. That's not saying chemicals are mutagens. That's saying some chemicals are mutagens. And this is a good time to remind everybody, because I haven't said it since chapter 2. Everything's a chemical. So when someone if someone's trying to sell you something, some hippie garbage, because she's in a pyramid scheme and says, oh, this is chemical free, right off the bat, that should be a red flag. I'm sure she's not lying to you on purpose. She probably thinks it's chemical free, but a chemical is everything. Water is the chemical. So if I'm giving you water and saying that's chemical free, that's a lie, because it's filled with dihydrogen monoxide, which is water. Anyway, any questions about mutagens or mutations and how they occur? All right, your book also points this out, as I've been saying. Mutations are often harmful, obviously, like sickle cell disease, harmful. There's an exception to that. We'll talk about that in the next chapter. And sometimes they can be good, right? So it's mutations that are the source of the rich diversity of genes in the living world and the diversity that makes evolution by natural selection possible. So again, if that one moth didn't have that one mutation, there would be no black moths to start with, right? And there's only a few, maybe even one to start with, that one black moth that was hatched. That thing had an advantage, and because of that, it outlived the light ones and had more offspring. And then those offspring, again, had an advantage. So they lived longer and had more offspring. And then their offspring, right, so on and so forth. So generation after generation, we had more and more and more and more black moths and fewer and fewer white moths. Why? Because that one moth had one mutation that changed the color of her offspring. So anyway, any questions about that? Now the quick and easy part, I think. Again, this chapter is really useful, in my opinion, even for non-biology majors, because to understand how viruses work and understand how vaccines work, not all of them, but largely, you need to understand everything in this chapter. So when you are confronted with a situation like, do I need to give this vaccine to my kid? I know that there's a 0.002% chance there might be some sort of vaccine injury. I know there's a 17% chance that they're going to catch this disease if they don't get it. And you're trying to weigh the options and you want to do some research, and I'm using quotation marks because it's really not research. You're really just reading other people's research. But anyway, when that happens and you're trying to figure out what you want to do in your life, then you're not starting from scratch, right? You're going to have an understanding of the basics. Anyway, let's get into it. Viruses, what are they? Well, first of all, they are the next attendance word, virus. Viruses, I don't care. You do it plural or singular. Anyway, viruses share characteristics of living organisms, but like we said back in chapter 1, they're not technically alive. So let's talk about the similarities and the differences. How are they the same? Well, they do have genetic material in the form of nucleic acid, the same kind we have, and they are packaged in highly organized structure. Remember, so those are some of the properties of life. But they aren't alive. Why? First of all, they're not cellular. And second, they cannot reproduce on their own. Viruses cannot reproduce on their own. Viruses, and I'm about to go into detail on this, but if you remember anything about viruses, it's this. Viruses basically hijack your cellular machinery. All they want to do is make more copies of themselves, and they can't do it, so they hijack your cellular machinery. Because as you know, in a sense, the ribosomes are essentially 3D printers, right? They don't know any better. You give them a code, they're like, all right, bring me this amino acid, bring me this amino acid. They're just building proteins. If you give them the code, they will build the protein. Right. Huh?
What is the word you said? Hijack.
Hijack, yeah. I mean,
No, what did they hijack?
hijack the ribosomes, the cellular machinery. Of course, I'm not going to ask it like that on the exam, so you don't need to know it verbatim. But what I'm saying here is the viruses take over your cells. They take over the machines. As your cells produce proteins, well, the virus needs you to produce their proteins. So any questions so far? We're still going to talk more about viruses. But the viruses are basically infectious particles consisting of genes in a box, right? Because they don't have any of the organelles. They can't take care of themselves, but they're just the instructions, right? They're just basically instructions on how to build them. Nucleic acid wrapped in a protein code. In some cases too, they also have a membrane. I'm gonna put an X to this, not because you don't need to know it, but because you don't need to know it for the exam. So basically what we're saying is, when we think of our genes, right, we have chromosomes, and our chromosomes are in the nucleus, right? And the nucleus is a membrane, right? That's what the nucleus is. And some cases, viruses have that, right? They have the genes, and those genes are wrapped in a membrane. And sometimes, probably more often, they have the genes like we do, but instead of having it wrapped in a membrane, they're just covered in a protein coat. And again, for the exam, that's not that important. Actually, now that I'm thinking about it, most of the information I'm going to give you for this chapter, you could probably not even know this, now that I'm thinking about it. For us, at this level of learning, I guess that is trivial. Anyway, like we already said, viruses can't reproduce on their own. So because of that, they have to infect living cells and direct its molecular machinery to make more viruses. So that's what I said earlier, right? Just in a different way. Cells hijack the cellular machinery. That bullet point says the same thing in different words. Any questions about that slide before I show you a generic picture of a virus? Okay, I need to move forward. I'd love to finish this chapter by today. Anyway, there it is. There's a protein, a virus. They're all different, so this isn't, not all of them look like this. But here we're just saying basically, right, there's genes wrapped in a protein coat. In this case, the genes happen to be on DNA. It's not always the case. We'll talk about that here in a second. So there's different types of viruses because they attack different things. Any idea what bacteriophages, that's a type of virus. Any idea what they may attack based on their name? It's not a trick question. Bacteriophages, what kind of virus? Those viruses, what do you think they attack?
Bacteria.
Bacteria, correct. It's all in the name, right? Viruses that attack bacteria are called bacteriophages, bacteriophages, people pronounce it differently. Basically, it means bacteria eaters. They consist of DNA enclosed in the structure of proteins. This is actually what some of them look like. To me, I think it's really interesting. That's all mostly made out of protein. It looks like a little space capsule, but that is what they look like. Here's an actual microscope picture of one, or excuse me, 3 of them. You can see 3 of them on the outside of the bacteria. This one's injecting its molecular, good grief, its genetic material, right? The DNA from the virus is coming out and going into the bacteria.
And it's like that word once.
Yeah, it does. Here's a better picture, I think. Of course, it's not a real picture. It's an artist's description or whatever. So they almost look like little hypodermic needles, and in a sense, they are. It's attacking this bacteria, which just happens to be an E. coli, and injects its genetic material. That thing then injects itself into the bacteria's chromosome. They only have 1 chromosome. And then, the cell does the rest, like we already learned. The RNA polymerase reads the code and makes mRNA. And then the mRNA is then translated into different proteins. And then those different proteins come together to make a new virus, and it keeps doing it until there's not enough room, and the cell explodes, and all those new viruses escape, and then they go on to attack more cells, and then the cycle continues. But let's make it a little bit more complicated. There's 2 different versions of what I just described. There's the lytic cycle and the lysogenic cycle. For studying purposes, I hate to do this because I prefer you to learn it instead of learn how to get the question right on the exam. But in short, you can remember the lytic cycle is the short cycle, and the lysogenic is the long cycle, and it's in the name, right? The lytic is a shorter word than lysogenic. So if you remember that, then you can remember the lytic cycle is the short one, and the lysogenic cycle is the long one. And I probably won't even ask you questions about that. I'm not sure yet. More importantly, because you guys aren't biology majors, and because you guys aren't made of bacteria, I consider this a very unimportant part of the chapter. So I'm going to kind of go through it pretty quickly. You can download that PowerPoint and watch this in action. You can also download download the PowerPoint, watch this video, see it in action. Here's what the lytic cycle looks like. Where are we at here? Step 1, like we've already seen, right? The virus attaches itself to the bacteria and injects its genetic material. Now that genetic material is hanging out in the cell with the original genetic material. And then again, the RNA polymerase does its job. It reads the DNA, it copies it to a strand of RNA, That RNA is then translated into some proteins. Those proteins combine to make a bunch of new viruses until there's so many that the cell just explodes. And then those viruses are out to go infect other cells, and the cycle continues. Pretty simple, right? That's the lytic cycle. And I'm definitely, like I said, I probably won't ask you any questions about this, but I'm definitely not going to ask you which stage 1, step 2, step 3, step 4. You're not going to have to know all that. All right, so that's the lytic cycle. Let's talk about the lysogenic cycle. This one's a little bit more complicated, a little bit trickier. So it looks the same in the beginning. The virus attacks the cell, the genetic material goes into the cell, but here's where it gets a little bit trickier. Now that genetic material, instead of just hanging out on its own, it now basically fuses with the original chromosome. So now you have the chromosome with the bacteria information, the virus information also in it. Also, this happens to humans, and it's really fascinating, and the way we see things different than other humans or other creatures has to do with this, but we don't have time to talk about it. Anyway, so because now that DNA is incorporated to the bacteria DNA, and it hasn't been read yet, right? So haven't even done transcription. It's just incorporated itself into the cells, the chromosome of the bacteria. So now, when this thing goes through mitosis, it's just going to make a perfect copy of it. So now every new perfect copy of this cell, of this bacteria, has that viral DNA in it, right? And all the virus had to do was insert it once. And then the bacteria just did the rest of the work for it because it just copies. It makes a perfect copy of itself in mitosis. So that's the first part of the lysogenic stage. And this is why it's longer, you see, because it doesn't kill it right away, right? It inserts the DNA into the original chromosome, and then it makes a perfect copy. So then you have millions of cells, millions of bacteria cells that now have that copy of the virus DNA. But then eventually something triggers it, and then, that's when transcription and translation happens. So all that information that was in the DNA is then translated into some proteins, which are then built into viruses. And then there's too many and it explodes and the cycle continues. So basically, lysogenic is basically when the virus attacks and the information is incorporated into the original chromosome and it's copied and copied and copied. That's all that happens, right? It's not considered the lytic cycle until basically transcription and translation happen. Any questions about that relatively unimportant chunk? All right, the same can be said for this. We aren't plants, so I'm gonna go through this kind of quickly. I probably won't ask you questions on this. Also, it's really simple, at least the way the book teaches it. For plant viruses, let's just say they're usually not deadly. For your own knowledge, I'm not going to teach you on that. But they usually just stunt plant growth and diminish crop yields. I guess that's why it would be useful for us, because even though we're not plants, we do eat plants, right? Your book points out that most plant viruses are RNA. Your book also gives an example, which is slightly important, not for the exam, but the reason it's important, well, you'll see. Many plant viruses, like the tobacco mosaic virus, are rod-shaped, like you can see up there, with a spiral arrangement of proteins surrounding nucleic acid. It doesn't just attack tobacco, it also attacks related plants. And the reason I think your book even talks about it is because this is the first virus ever discovered. It's the TMV virus. I'm not going to ask you any questions about it. Here's what a tobacco leaf looks like. It's been, disinfected, which of course probably doesn't matter to you unless you know what an actual tobacco leaf looks like. But there it is. Here's the virus itself. And now we're going to zoom into this little portion right here. And you can see the RNA is all coiled up in there, and the protein is around the outside of it. Again, this is unimportant for you guys because you're not plants. So we'll go through this quickly. I'm not going to ask you any of this. But the plant virus has to get past the plant epidermis. So the plant has a way of protecting itself. But because of that, therefore, if you have a damaged plant, it is more likely to be infected. So the people that do trimming of the trees, if they're doing it right, they have to know what they're doing. Otherwise, they have a chance of actually infecting a tree with a virus. Because when you cut things off of it, that's a wound, right? That's a plant wound. Also, farming tools can spread viruses. Because if you cut one plant that has a virus and then you go and cut another one, then you spread it. The book also points out that there are no cure for most viral plant diseases. And because of that, agricultural scientists focus on prevention and breeding or genetically modifying varieties that just don't get the infection to begin with. And I wish we had more time to talk about it. That's a relatively important bullet point, not for the exam, but people are all afraid of GMOs, but they don't properly understand GMOs. I can see how some of them are scary, but some of them, you could literally just take the genome from the plant and say, whoa, the viruses attack that gene, so why don't we just change that gene so the viruses can't attack them, right? That's not going to affect us as the people that eat them, generally speaking. Anyway, let's get into some important ones here. Important for you because you are an animal, right? Humans are animals. So let's talk about the more important stuff here. For animals, obviously viruses are common causes of disease. We know that. Right now we're in the, I don't know if you guys have seen the news, but they're calling it a triple pandemic, I think, because we obviously still have COVID, we have flu, and then we have RSV. So it's really bad, really dangerous for little kids. But anyway, we don't have time to talk about that. Your book points out that there is no greater health threat than the flu. And you can look that up. Right now, I think COVID is still surpassing flu, but that's right now. But if you look at the history of mankind, according to your book, and I still think this is correct, the most deadly virus ever has been the flu. Now that might sound weird to you because you might think, well, I had the flu and I survived, or most likely, I know a lot of people who've had the flu and did not die, right? But if you're looking at the grand scheme of all human life, the flu has killed a lot more people than polio or HIV. And again, this is a great topic for independent work. It's different than saying it's deadly because if I get it, I'm likely to die. We're not saying that, right? Because like if you get Ebola, you're probably more likely to die than if you get influenza. But you're more likely to get influenza than you are to get Ebola. So it all depends on how you define deadliest. Anyway, like many animal viruses, flu has an envelope phospholipid membrane with spikes of proteins. As far as we're concerned, that's not very important because we don't get into details of that. But basically, it helps because of its membrane. It helps merge with our membranes, and that's how it affects us. But at this level of education, we don't need to know that. Many animal viruses have RNA. Some of them have DNA. You don't need to know which are which for the exam, but your book gives some examples. For RNA, we have flu, common cold, measles, mumps, AIDS, polio. For DNA, we have hepatitis, chickenpox, herpes. For independent work, you can look it up and tell me what about COVID. What? And if you really know, no, I'll just go ahead and tell you. The COVID, right? What was it that we were calling it at first? The coronavirus. Coronavirus is like a family that includes the common cold. So basically, the COVID is a mutation of that common cold. It's just a lot deadlier. But I remember when it first happened, the conspiracy theorists were like, they knew this was coming, and they showed pictures of like the Lysol wipes, and it says on the label like kills 99% or it kills these viruses. The coronavirus was listed. It wasn't. See, I told you this is all a plan. They knew it was coming. But had they had Bio 101, they would know that a coronavirus is basically a family of viruses that includes a common cold. So yes, we've known that for years. So yes, we've been fighting coronaviruses in general for years. Anyway, any questions about this? As far as the exam is concerned, that's not important. I'm not going to ask you which is which. Here's a picture of one. This is kind of interesting, right? It looks like a plant virus, like a bunch of plant viruses, but in a membrane. Not that that matters. I'm definitely not going to test you on what they look like. Let's see one of them in action. They all work different ways, but this one happens to be a, what does it say? Which one is it? Reproduction cycle. I don't know. I guess this is just a generic thing. I was hoping it would be an actual virus, but this is just a generic flow chart. So here we see the membrane from the virus is merged with the membrane for the human or animal cell. So now the thing comes in, right? Then we remove the proteins to get to the genetic information. We have an enzyme that is included in the virus, so it can read its own information at first. It reads the information and makes some RNA, and then that RNA, half of it is used to make new proteins, half of that RNA needs to go back into the new virus, and then the new viruses are made, and then they explode the cell and go on to infect another cell, and the cycle continues. So it's very similar to what we looked at with the bacteriophages, right? Infects it, You make new versions, and that's that. And for the exam, you can see all these different numbers. Number 4 is protein synthesis. Number 5 is RNA synthesis. Number 6 is assembly. There'll be absolutely no questions about that on the exam. Here's the picture that shows it all together. The next word for attendance, we'll just say is mRNA, since we have it up there. But anyway, the last 2 pictures I just showed you, this is them together in one picture. If you download the PowerPoint, you can watch this video that shows everything I just showed you, but it's animated. Here's a picture of the mumps virus. You can see the protein spikes. You can see the nuclear envelope. Your book points out, and this is really important, that not all viruses reproduce in the cytoplasm. So everything I just showed you was happening in the cytoplasm. Here's where things get a little bit trickier and slightly more dangerous depending on how you look at it. The herpes virus. If you've ever heard, it's called the gift that keeps on giving. And the reason why is it's not like it gets inserted into your cell and then we start doing things right away and make the cell. No, it's almost like the lysogenic cycle from the bacteria. What happens here is it actually incorporates itself into your genome, right? So then it's there always. Every time you go through mitosis, you're making a perfect copy of that virus, right? So then, yeah. Anyway, if any of you have ever had chickenpox, you are at risk for shingles. Basically what happens if you have chickenpox, again, it incorporates itself into your DNA, and then later in life, something triggers it, and then it expresses itself as shingles. Can look that up for independent work. It's pretty interesting. We won't get tested on it. Let's see. Yeah, that's basically what I said, right? Herpes virus incorporates itself into your genome, and then later in life, things trigger it. Could be, and then also it doesn't have to be shingles. It could be like a herpes virus like we think of it, like a mouth sore or something like that, right? Anyway, I'd like to tell you more, but we are out of time. Actually, yeah, we are. So I'll see you guys on Monday. Have a great weekend. If you feel like dressing up for Halloween on Monday, feel free. I'm going to be dressed up as a tiger professor.